basically have started this series of webinars in the context of basically in the context of lockdown, but then we took this important topic related to COVID-19. So we have first seminar last week by Dr. Iqbal, who talked about generally about the preparations Kerala government has done to contain the COVID spreading or all the other things. So now we have today Dr. Gowri Rajam, who is basically a associate principal scientist with Merck in USA, and he is an established scientist with a large number of publications in various international journals. And from 2003, he was leading a team of researchers at the Immunology Laboratory, Center for Disease Control and Prevention, Atlanta, and pursuing research in vaccine development and evaluation of pathogens of novel public health interest. So I believe that he is the right person to talk today about the current topic, and his experience and his expertise would be much beneficial for the viewers and the audience. So I don't want to take much time. I thank the organizers to this webinar who has taken lead in organizing it, and it's a part of the series which will be continuing for the next 3-4 weeks also. So I request Dr. Gauri Shankar to take over and then continue with the presentation.
Thank you, Dr. Sudhir, and thank you. Sarika was the reason for me to be here, so I should thank Sarika and thank Arjun for the dry run, so that I can just. I'm not technologically savvy. I've got a very limited technological. I still believe in blackboard teaching. I believe myself as a teacher and researcher, and I still believe in blackboard teaching. I do miss my blackboard teaching days. So when Sarika reached out to me with this opportunity, so like, this is great. I didn't even ask what exactly the mix of participant. I just jumped onto the occasion because I have moved away from teaching a bit. I do teach. I have a team of researchers in my lab, and whenever they just ask me a doubt, they get it. They immediately I take them to the conference room, dry erase board, and I just teach them. So I do. My teacher and me just get the feed every now and then. But this is a banquet, so I would not miss an opportunity here. So thank you very much, and I would like to till last week, I would not be really just I was not keen on talking about COVID because like my interaction with COVID or coronavirus was very limited because Merck was not really actively involved in, at least officially, Merck was not actively involved in either therapeutics or vaccine manufacturing for COVID. So I was pretty much behind. I was not doing much of activity. But now Merck into the vaccine ship. So we are working on two vaccines for SARS-CoV-2 and one therapeutic. And I am sort of slowly involved in the activity. So I feel like I do have some merit to be within this group. But for that, let me just give you a disclaimer. I know very well there's so much of information out there, and almost all of you, especially the younger generations, like young students, they're all so much like technologically savvy. I am pretty sure you guys have a lot of information about either the virus or the pathogenesis of the virus or all the nuances of the viral biology of this infection. So I am only going to scratch. I'm only going to scratch the surface. My intent is basically to just number one, keep the trigger people's interest in this virus or the infection. Number two, maybe set straight some of the facts about this virus and the infection. But so it is possible that some of the information or many of the information may sound redundant. So if there is a case, it's not my fault. It is actually the fault of the information overload and social media. So with that disclaimer, let me just start my presentation. And the topic of the day is I will try to give you a flavor of this the pandemic. I would like to just like take you and get to some like I will try to just take you on a tour to see both side of this virus. Trying to giving you a sort of a bird's eye view or a flavor of how the virus interacts or what exactly the virus sees in the host, and also we also see how the host sees the virus. So how exactly the virus sees, what exactly the virus sees, and how exactly it just like reacts to that like a visual and physical interaction, and how exactly the host tissues like a response to that physical interaction between the virus. That exactly will be my narrative. Okay, I know this is not a classroom, so you cannot really interrupt me. I wish if it is a classroom, you can just interrupt me, and of course, if that's the case, then this discussion may go on for a couple of hours. Mercifully, this is going to be a short presentation. At least for you, it will be mercifully short presentation. Okay. Before going to it, we have been. I'm pretty sure, like, if not for all of you, at least some of you, it will be like we have been hearing about pandemic, endemic, epidemic, and all those different this generation is the sort of explosive generation of omics. I'm not going into this genomics, metabolomics, all those omics, but I'm just saying like all in the public health, we don't just hear so many omics. I would like to just set the stage for people to understand like when do you call which one you call as endemic, which one you call as epidemic, which one you call as pandemic. A little bit of like a basic public health 101 before we jump into the actual current pandemic. So bear with me for maybe for a couple of minutes to just set the stage, okay? Okay, endemic. Let me start with a smaller area, endemic, because usually endemic refers to the disease that is confined to a community. A classical example of endemic is filariasis, which is pretty much has got a very small footprint. It usually just like occupies very small places. Like, of course, that doesn't prevent people from moving around. Pondicherry is supposed to be endemic for filariasis for a long time, but it never prevented people from Chennai to just go to Pondicherry for obvious reasons. We have a lot of interest in Pondicherry because you know very well that Pondicherry is a basic union territory where the liquor prices are very low there. So people used to just go there for that. Not only for that, but like, hey, that's attraction, especially for teenagers. Like I have done it. Okay, let me be honest about it. So Pondicherry has got its own attraction. We'll be so scared when we go there. We'll be so scared about the mosquito fight, but still we'll go there. So that's about endemic. But even the endemic, you can basically you can separate them into a sporadic and also hyperendemic. Depends upon the frequency of occurrence and depends upon the intensity of occurrence. A classical example of what's like an endemic is very sporadic polio. Like I'm pretty sure, like as a Indians, we should be very proud that we managed to just eradicate polio. Like earlier, like maybe 5 years or 7 years back, like polio was sort of zeroed down to four countries. That's why they are called as pain countries: Pakistan, Afghanistan, India, and Nigeria. But now, pretty much like India is out of the picture now. Now it's only a pan countries, and hopefully with all this pulse polio, all other efforts, hopefully we'll be able to eradicate polio. But even though technically polio is eradicated from India, still, what, every now and then you would just see in social media that like Rajasthan or some other place we have just we have a case of wild polio. So it is very sporadic. It happens, but pretty much in the case of hyperendemic, classical example is malaria. It is still there, but it is certainly confined to small pockets. Actually, you could see this when you look at the list of like a cases. I mean, in some places it is very high. Some places that is basically hyperendemic, where it is sort of confined to a specific region. So this is exactly how we just define. So when you hear about endemic, when you hear about basically like sporadic, etc., I'm pretty sure you will be able to really just connect your understanding now. Epidemic. So next, like, we are actually expanding the geographical impact zone. from endemic, where this expanded into epidemic, where it basically expands over an increased amount of like a disease in population. A classical example is dengue, where basically you can just see that when it happened, it sort of swept across the country. It didn't discriminate, it didn't differentiate people. It just sort of swept the country. Um, but of course, where we are now, we didn't hear about pandemic 10 years back. Pandemic was basically more on a history, but now in the past 10 or 15 years, we are hearing more about pandemic. Like the SARS was a classical example of pandemic of our age, MERS, Ebola. Ebola was basically pretty much it was threatening to be a full-blown pandemic. Fortunately, we were saved. And I'm pretty sure Merck also need to be given a credit because Merck developed vaccine without any profit. Merck developed a vaccine and gave it, and that basically was able to just sort of like confine the Ebola to a small region. True pandemic. You can just see basically where COVID impact. You can see that it has not spared its continent. It has not spared any country. Earlier, we thought like tropical countries are spared, but later we realized that it doesn't spare any country. COVID is COVID is a true example of a true pandemic. Where did it start? So this is basically I'm telling you this slide. Any one of you here can actually run the slide. We know so much about this and this step. It is like I have not seen it anywhere. That's why I don't want to put it there. And unfortunately, the market is open now. Again, so I don't know what's going to happen in the near future. But one thing is very interesting is that within a matter of like over 2 months, from the start, that is 30 December to 18th February, we could see that they basically we know both ends of the disease. We know the exact signature or blueprint of the virus. We also know what exactly was this eukaryotic receptor, host receptor. We also know the exact information about the structure of the receptor. Great. That is exactly is where it shows how far we are, how ahead we are in scientific technology. It's growing. It's basically just the last week. I just pulled it out last week, and you can just see that it is increasing. It has like impact on almost 5 million people across the globe, with almost like a 350 fatalities. It is growing in India. Like it was not like this. It is growing in India, and obviously more tests being done. I'm pretty sure like more we'll have more information. I hope this rate remains low. That's all my hope is that as far as the case fatalities is low, it doesn't matter. Like if you happen to get exposed, you are going to get you're going to get a natural immunity. But you can see here in US, in Western countries, US, the impact is very high. We are having more fatalities. Obviously, because we are so immunologically, we have been cocooned. Now, when we are getting sort of hammered by this virus, I think our immune system is taking time to wake up. And before that, if you are young, okay, you are able to just tolerate it. Maybe the elderly, they have no time for responding. And you know very well, this is a generation of baby boomers. So we have a lot of people in their 70s and 80s. That's the reason why we are seeing more case fatalities in Western countries. Cool. Okay, let us just move on to the biology of this virus. Standard RNA virus. I'm pretty sure any tech would just show you this. Very standard RNA virus has got a very. It looks so nice. It looks so beautiful. You see, like a thin cushion. Like it has got a core where the RNA is very nicely packaged the nucleocapsid protein so that it is protected very well. And then you've got a lipid bilayer that basically just covers the entire package. And I'm pretty sure it is lipid membrane. I'm sure the biologists or biotechnologists would be able to just visualize the beautiful design of Danieli and Dave's and right the fluid mosaic model and all those stuff. Yeah, hopefully you will be able to recall the fluid mosaic model of lipid bilayer. It is universal. Whether it is a viral membrane or whether it is going to be eukaryotic or prokaryotic membrane, it's the same. It is a beautiful phospholipid bilayer membrane. Then it has got the real hero here for the virus, not for us. for the virus is the spike protein. This spike protein, like the tissue-like protein, this is the one that is actually going to just like, it's actually like an antenna. This is the one that is going to just recognize the specific host sequence and just avoid. It is like a docking. It's like a docking, like a antenna. It goes, docks into the host receptor, then basically triggers the host receptor, and then basically things happen. So I think we are more interested on spike protein for pathologists or viral pathologists to understand how exactly what is the mechanism of the infection and invasion, and for the vaccinologists to see how exactly we can exploit this protein to just develop like a vaccine for that. Because ultimately, this is the outermost component of this virus that is being exposed to your immune system. This is what your immune system is seeing. This is what your immune system is trying to recognize. This is what your immune system is trying to memorize. And this is what your immune system is trying to recall, is the same virus again. So obviously, as a vaccinologist or as an immunologist, we are also interested in seeing which of the spike protein, which form of spike protein is actually is very immune dominant. Which one can be exploited as a vaccine? So that's exactly is our interest. Little more information about this virus. The coronavirus, you know very well about that. It's corona because it's like a crown and like a single-stranded positive sense RNA virus. Okay, we know. And there are 4 groups: alpha, beta, gamma, and delta. And I have just color-coded alpha and beta because they are the one that is actually the animal viruses. And honestly speaking, we are unnecessarily blaming the SARS virus for all these activities. It has got SARS virus has got no interest to infect human. It was happy it was living in bats. We unnecessarily take, who asked you to go and eat the bat? Who asked you to go into the cave and basically disturb the bat population? Then you keep just like a deforesting. Then obviously what happened? You are just like you're just messing up with their natural habitat. When the natural habitat is affected, where will the bat go? The bat will come to your house. When bat comes, it is not coming alone. It is coming with its own like a native flora. And the native flora basically is when bat population is going down, the native flora is evolving. Normally, what's going to happen basically? Virus is evolving. So you are going to have like when there is going to be variants of this virus. Some of them may have a spike protein that no longer has got like a specificity for the bat receptor, but it does not have an alternate host. So it just basically goes off. It dies. Whereas if you disturb the habitat, if you happen to be in your house, when the virus basically when during evolution, if there is going to be a mutation in its spike protein, wow, I have Sarika here, I have Arjun here, I have Shankar here. Now I got it. So it is we who brought them into their house, and now we are blaming everybody. So it is a human. I think it's our culture. I think our living style that is now actually predisposing us to more pandemic and more public health challenges. I think people need to just realize this. Nothing wrong in basically taking one step back. Sometimes it is good to just take one step back to just move forward. And that's exactly what we need to do as far as the food habits or as far as the way in which we basically look at the wild animals. Same thing here. See, like these are all like a SARS virus, MERS virus. They're all basically like pretty much they were very happily living after in bats. Why this CoV2 became a problem for us? Because it has got a mutation in the spike protein, where because of the mutation, it has sort of lost its specificity to the bats or rodents. What's going to happen? It has got no alternate host, so obviously it would have just confined itself to bats and it would have died. But now, but during this mutation, it has higher affinity for human or pangolins or receptors. Okay, so pangolins are there. We love pangolin. At least Chinese love pangolin because they feel like it has got a lot of medicinal properties. So people just butcher pangolin, take out the scale. During the butchering, they're not going to wear a mask. They're not going to have a PPE for doing it. It's the open market butchering. So the virus is going to be spread all over the place. Inhale it, pass it on to your brothers and sisters. That's what is happening. So this is exactly what I was just alluding to. It's like it's we say origin because humans we are ready to blame. We never used to assume, own anything. We don't own any blame. Moment there is something happen, something going wrong, we would try to find a reason for that. So immediately we say origin. Bat is not an origin. It is a natural habitat. E. coli in your stomach is not an origin. E. coli is a normal habitat. If somebody is going to basically set up like wade into the sewage and basically say like, oh, human is the origin, that is nonsense. You should better know that. If you're going to just wait and do it without proper protection, you're going to get it. That's going to happen. Same thing here. So basically, like what happened, as I told you, because of the mutations, they were able to find the intermediate host. And when they happened, when humans happen to just come across, then basically they become the alternate, like an alternate host. Especially in the case of SARS-CoV-2, primary host is bad. I don't want to say that, but like that's what the literature says. I'm going by that. The alternate host is actually pangolin, and either the direct contacts or indirect contact, either the bat or the pangolin, what exactly happened, human host gets it. And then human basically there's a human like person to person transmission. Like social medias are doing very good. They're not helping us here because social medias are doing a very good job of distancing ourselves from each other, right? So even the restaurant when four friends go, basically what they do, moment they go in, immediately turn on the phone, either they share their like images or start working on the WhatsApp or Facebook, etc. So they are very much. I think one excellent example, one excellent like mechanism strategy to enforce social distancing is like restaurants should enforce that. Hey, whoever comes in, you should just see your phone. Beautiful. Then I'm going to be like 6 feet away from my friend. But unfortunately, that's exactly what happened. When there is a human-to-human interaction, that's a phenomenal spread. That spread, like coronavirus, that spread. We assume that it is 6 feet, but it is much beyond that. So at least 6 feet. That's why at least 6 feet distance is being advised. Most of us don't even feel we have the infection. Some of us basically may succumb to the infection. They end up in the hospital. So this is exactly how the transmission takes place. So at this point, let us all like as a scientist, let us take a resolution like let us ban live animal markets that trade wildlife. I'm not saying no chicken, no mutton, no fish. Wild animals. I'm telling you. I like chicken 65, so I will not agree with that. But wild animal, let us basically stop wild animal. Like I was reading an article yesterday. I think in India, right? Where India or Kerala, I don't know where they just found that the elephant basically like ate like a pineapple with the firecracker there. And I believe like the pineapple was usually that's the way they do it to just like kill or distract other animals, which is again law. It is not. It is like it's unlawful. But again, elephant doesn't realize that there was a firecracker there and look at it, what happened. And apparently it's because of the poachers, right? It is a part of a poaching. So people need to stop buying that ivory. Like when we go, we need to stop buying ivory. When there is no customers, and obviously they don't have a product. We do that. That's a problem. So I think we need to. I think it has to just come from us, and hopefully, like we can just take a resolution that like we will not buy anything made of wildlife, so that like market shrinks, that discourages focus on manufacturing. Okay, that's.
Uh,
Can you?
Yes.
Now it's getting loaded, sir. One minute, sir. Yeah. Now it's now we can see.
Technology, I don't know what's happening. I think this cartoon, I'm pretty sure every one of you would have seen it, especially for people who are in science. Moment the COVID hit the news, I'm pretty sure you would have Googled it and you would have just seen this cartoon. Very popular cartoon. What I want to do is I want to just bring your attention as a like a microbiologist or as a science folks. I want to draw your attention to these three components: the ACE2 receptor, the protease, and the spike protein. As far as the receptors are concerned, these receptor ACE2 or angiotensin converting enzyme to receptor, they are expressed in like epithelium, heart, like expressed in heart, lung, kidneys, and GI tract. And they usually the ACE2 receptors usually come as a package. This enzyme usually just like we've seen associated with receptor. And this is the receptor which is recognized by this protein. Please understand that it is not that the SARS-CoV-2 virus basically wants specifically to recognize the receptor. It is just it's a game. It's a permutation combination where it so happen these like two different like a receptor as well as the molecules like this three-dimensional structure seems to fit like a lock and key, hence they just came together. That's all. It is like I'm a strong believer of Darwin. I don't belong to Lamarck. I'm a strong believer of Darwin, where it is a part of evolution. When you happen to just come across, we happen to have a best fit, it fit. No fit, you don't fit. That's exactly how it has happened. It's all, see, what is scary? What is scary is, which means it is a game of chance. 1 in 10,000, 1 in 100,000. Like if there is going, if we have a modeling folks around you, like a biological modeling, not the real modeling, biological modeling or modeling folks around you, they would say, what exactly when you plug in the algorithm, you can see what exactly the frequency of having this chance occurring. It is like could be a 1 in million, 1 in 10 million, but still it occurs. And when it occurs, it can be devastating. That's exactly what is happening. What is the chance of a viral protein, virus undergo, where its spike protein is going to find as going to recognize a ACE2 receptor on heart, lung, or kidney, or GI tract. But it happened. That's why we are here now. So when we just like take a quicker, closer look at this, like what exactly is happening there? This is a very complicated interaction. Just I want to just draw your attention. I don't want to take much of your time. I want to draw your attention. Like basically, what the enzyme is doing is enzyme is actually truncating this ACE2 receptor. It is actually just chopping off the like a. It's almost like this receptor ACE2 receptor is capped. So it is removing the cap, exposing so that the interaction between the ACE2 receptors and the spike protein becomes much more stronger. And this truncated receptor is actually stabilized receptor now. So this interaction between ACE2 receptor and the spike protein is more stabilized. And this, please see, if there is no truncation happening, then basically the spike protein may just attach to the ACE2 receptor, but will not undergo uncoating. May not just get into this cell. But unfortunately, the protein, it truncates, makes it more biologically active, and this active interaction between the spike protein and the receptor results in basically like entry and uncoating. Why it is happening? It is a normal responsibility of the ACE receptor, because this is basically is one mechanism of actually endocytosis. That's what it is doing. It is doing its job. But unfortunately, instead of a normal individual, if there is going to be a sort of like an abnormal individual get into my car, what can I do? Driver doesn't care anything. Driver basically is supposed to say, hey, if somebody is going to get into the car, drive them from A to B. So I'm basically a robot driver. That's all it is. It's like when you are going to have a driverless car, that's what is going to happen. It is voice activated. Basically, I record your voice. I just get into your car. It's like, hey, take me to this place. It doesn't care. Car doesn't care. Car is not going to turn around and say, who is here? So it's going to take. Same thing happens here. So that's exactly what is happening. Where with this inadvertent, I'm telling you, I am a strong believer that we cannot blame SARS-CoV-2 inadvertently. Our system actually facilitates the entry and establishment of SARS-CoV. Again, SARS-CoV-2 has got no interest in the court of like immunology. I would just represent SARS-CoV. Okay, you may all represent you host. I would represent host 2, and I would say, Your Honor, I am being unnecessarily blamed. It's not my mistake. I am undergoing a normal evolution. I happen to come across a suitable lock. I'm just coming here. Why is that the lock is actually opening the door? lock and just keep locked. Like the membrane protease is actually opening the door. So I'm just going, hey, come on, man. You cannot stop me. That's what is happening here. And he's very smart. Once it just comes and say, obviously, what's going to happen? He has a positive sense like RNA. That's great. And it has early genes are actually which are translated or replicase. Wonderful. So moment replicase enzyme is translated, it is going to facilitate the replication of positive sense to negative sense. So basically, like more negative sense, more mRNA is generated, more translation. Wonderful. On one hand, you are actually producing with the help of replicase, you are producing more and more of positive sense RNA, as well as more and more of mRNAs. That's actually going to give more proteins. They are all translated effectively, packed. Again, how about host? It doesn't care. It is non-specific. It doesn't care whose information is there. As long as mRNA is there with appropriate signature, it is going to translate. It is going to produce the proteins, and those proteins are going to be packed. And obviously, because the outer membrane is actually a lipid membrane, it is going to just set up. It is very easy for it to basically fuse with our own membrane. That's the reason why when you look at the evolution, I'm pretty sure we are trying to kill some of our ancestors. So I'm pretty sure these viruses are all our ancestors. That's exactly how the system would have joined. Few viruses joined to become a mitochondria. Few viruses joined to become a Golgi apparatus. But they are all supposed to have their own autonomous DNA, right? So a few mitochondria joined together to form a bacteria. Few bacteria joined together to form a small organ. So we are talking about our own ancestors, maybe like 100,000 years back. So obviously, at the end of the day, wonderful. Basically, new virions are formed. They just sort of very happily they just borrowed of the cells and they just look at the other cells. Look at this. This is the interaction. This basically the interactome just gives us a flavor of how exactly the interactions are. How many interactions are happening? Where the green circles are actually viral proteins, and the blue square box are actually a human host proteins. You could just see almost like 200 different interactions happen when SARS-CoV-2 just like enters the human. This much of interaction is required for an individual to just transition from a normal individual to a COVID infected person. So what is the value of this information? This information is very valuable because this is the point of research. A researcher can look at this diagram and say, like, hey, can I do something to just break one of the strands? Because you can see here, most strands are basically interconnected so much. If I have a therapeutic, I may have a natural herbal remedy. Who knows? I can have a natural herbal remedy which can actually break one of those interaction. Boom! If one link is broken, that means the entire cascade is shattered. We survive. That's what is going to happen. I think that is exactly the value of such the data available in the open space. Okay, we are not dumb. Okay, by the time I come by saying this, I'm pretty sure people will think like, wow, COVID is really cool. This fellow is really just going to create the havoc. Hey, we are smart. We have evolved so much. So our immune system is very much evolved. 80%, 90%, or 95% of the individuals, they have this beautiful sequence of response to the virus. Virus infects, be it a gut epithelium or lung epithelium, that's fine. They just come in. You have macrophages. You know very well. I hope you remember your old your immunology, basic immunology. Lung, you have your alveolar cells. That is, and in gut epithelium, you have got Kupffer cells. These are all macrophages. Hey, I didn't prepare those things today. Okay, I remember my basics. Okay, these are all like macrophages. They just scavenge. There are neutrophils. They are scavenged. So macrophage, PMNs, and the T effector cells. They play in synchrony. They play in synchrony very beautifully, handle the cells and activate the appropriate cytokines. Very, very responsible, restricted, controlled immune response. What is the end result? NK cells are activated. NK cells basically non-specifically kills these viral infected cells. B cells also participate. so that there is an imprinted memory there. At the end of the day, you have a T memory, you have a B memory, and effectively you're taken care of it. So 85% of the population, either asymptomatic or not, they don't even realize that they had a touch and go with COVID. Maybe only when you just look at the serum, all of a sudden you find out antibodies for this COVID. It's like, maybe you had a kiss with COVID, but for that you may not know because your immune system has very effectively handled this. But unfortunately, when you have some specific host factors, age, we can't do anything. You need to just, unless until we have technology to just tinker the telomeres, we have nothing else. We have, as of now, we have nothing to do. We have to just accept the age and move on. Okay? That's why I stopped seeing myself anymore. I don't want to see myself getting aged. Okay? Smoking. These are all things which we can control. Smoking, yeah, we can control. Diabetes, there is type 1, unfortunate. Type 2, we can procrastinate. But still, we can control it. We can control by medication as well as our sensible lifestyle. Like steroids, you can't help it basically. Like if an individual has got an autoimmune condition, an individual is undergoing a transplant, or individual is oncology, like a cancer therapy, can't help it. So these conditions is like almost like an elephant throwing a sand on its back, because this is going to open up an hyperinflammatory response. All these like cytokines like IL-6 or tumor necrosis factor, all of them, they actually like trigger over like a hyperinflammatory response, which is improportionate to what is required. Like for example, like these like you can just see here, like these cytokines are the real culprit for the immunological damage. I'm just going to give you only one component. There are several components, but one component that mediates the hyperinflammation, that is basically mediates the damage due to the immunological storm. That is called hyaluronic acid. This hyaluronic acid basically is like a gel. It's very good for you. Because like when there is a tissue damage, this hyaluronic acid basically forms a sort of a curve. It fills up the gap. It's like a gel. It's like a jelly. Like applying a sort of a Bonal or Neosporin ointment on the wound. But imagine your lungs are supposed to be dry. They are supposed to be dry so that they can if there is a damage there, if there is a production of hyaluronic acid in the lungs, so what exactly will happen? It is going to happen. It is going to make the lungs wet. It is going to make the lungs more like a wet blanket. That means it can no longer contract and just like no longer have the elasticity, but no longer have the physical characteristics to perform its function, and it does not have the biological characteristics because it no longer exchange gas. So you are having a compromised respiratory system. When there's a compromised respiratory system, you understand what's going to happen. When the oxygen is not transmitted throughout your body, the whole system is going to suffer, and that's exactly what happens. Where as far as COVID is concerned, because of that, it can have a very wide consequences. It can have a systemic disorders, which can range anywhere between just a simple fever, cough, to hypoxemia, or more profound is respiratory disorders. Because lungs, even though you may have a question in your mind like, hey, come on, man, these ACE2 receptors are expressed in heart, lungs, kidney, and GI tract. Why is that? More people are why is that the respiratory infection or respiratory conditions are more life-threatening? Because that is exactly is impacting immediately when you compare the other systems, and the impact is more profound. If there is going to be a GI tract impact, you are having a diarrhea, you are not going to die right away. Systems are going to still you are systematically you are okay. But if lungs are getting affected, then automatically what's going to happen is the oxygen, your brain is going to just send the message that, hey, I'm not getting oxygen, so I'm going to shut down. And that's exactly what happens in the case of acute respiratory distress syndrome, ARDS. It results in shutdown. So that's why it is quite a sort of a challenge, especially if you are having an highest group, then basically that could be an immunological overdrive. And if there is immunological overdrive, then basically that can predispose you to complications of COVID-19. Some of them you can avoid. Some of them you can, because like for example, vitamin B, vitamin B3. Apparently, it is supposed to be good and good to just support like a robust immune system. Similarly, zinc supposed to sort of support a good immune system. So if for normal individuals, if there is going to be some sort of a supplementation that is supposed to help, at least it would not hurt you, if not to help you. When you look at the like little bit of public health aspect of COVID, my question is the question is why it is a pandemic, why it is so contagious. Usually, when you talk to the public health like epidemiologists, they use this like a terminology called or not, that is reproduction number or not. Usually, like when you say or not, that basically that calculates the ability of a disease to be transmitted from person to person. If the or not is going to be less than one, usually the disease would just disappear on its own. We don't have to do anything. But if the or not is more than one, then basically it is going to be contagious. Interesting comparison. When you look at flu as well as like you see that the or not is very off. They are pretty much comparable. But why is that the COVID is or SARS-CoV-2 is more problematic? Because when you look at the persistence of the virus, this virus seems to persist in the environment much longer than flu. That's the reason why today, if you're having a flu, if you sneeze, you may have flu virus on the surface, and after like a few hours, you're not going to have it. So you're not going to be you're not contagious. You're not being infected. Whereas if it is COVID, then it is going to be there for several hours, even days. So even though person to person like our transmission number rate is pretty much comparable to the flu, unfortunately, because of its persistent nature, it can remain in the environment for much longer time and keep just infecting people again and again and again. That is the reason why the COVID-2 seems to be much more. It's a true pandemic now. Okay, I'm just moving away from like I don't want to just keep painting a bleak, like very painful, bleak picture. Okay, virus is great. Our system is little sometimes dumb, so we are not able to do anything, so we will suffer. No, I think when there is a challenge, we need to have a solution. Do we have a solution? Yeah, we have a solution. We have at least like a scientist have a strategy now. First of all, I need to know whether the individual is infected with COVID or not. So I need to have a robust diagnostics. Then I need to just make sure, like if he is positive, then I need to have strategies to treat him. Meanwhile, because there is pandemic, I also need to just like a kickstart strategies that can actually prevent, like at least protect those who have not contracted the virus. We just want to make sure like they are prepared so that if the virus happens to just contract them, they can actually overwhelm the virus. So three different aspects. I'm confining my diagnostics only to the viral diagnostics. I'm not discussing about zero diagnosis. To me, I feel zero diagnosis is not mature enough now. Because the disease itself was there only for the past 3 to 4, like 6 months. So we don't know much about the disease as far as the serological picture is concerned. Hence, we don't know much about the serological picture. It's too early to use zero diagnosis or as a diagnosis. Specialty has gone so fast. It has really just, like for example, using LAMP technology, isothermal amplification, we can just basically, we can detect this virus, or we can diagnose this within a matter of 1 or 1.5 hours. That's cool. So these molecular techniques are like, even though CDC specifically, even now, CDC like recommends PCR, traditional PCR, but still, we do have like diagnostic tools or diagnostic kits that use LAMP technology and have a faster diagnosis. Very effective. They are very effective, and they are already available throughout the world. Treatments. What are the treatment strategies? First line of therapy is alleviate the human pain. So treatment basically is the symptomological treatment. Treat them fever, cough, and all those stuff. Unfortunately, there is a one catch in this viral infection, because the target is ACE2, and it is known that some of the anti-inflammatory, like non-steroidal anti-inflammatory, seems to have a positive or seems to just predispose to a hyperinflammatory response. That's the reason why, even though traditionally, like Brufen, ibuprofen or all those non-steroidal anti-inflammatory drugs, even though they are recommended for, we don't have enough data to demonstrate. See, what is the in vitro or an animal model? They say, okay, when you take chloroquine, it is going to alter the pH or the intracellular pH. By altering the intracellular pH, it's going to impede the viral entry or propagation, etc. It is hypothetically, theoretically it is correct, but unfortunately that is going to happen. Then when I treat human beings with when I have two control groups, one treated with chloroquine, one not treated, the outcome is pretty much comparable. Then what do you say? What do you say about that? So chloroquine is still, it is there. It is being actively tested right now. A lot of studies are going on. We don't know what's going to be the outcome. This is one drug which has been approved for use. There is remdesivir. It is a nucleoside analog. It is basically, it is not that we don't know about the benefits of this, because it is a drug that is known to have antiviral. It has been demonstrated to have antiviral efficacy, because first of all it was used to treat Ebola virus, and we know very well it is effective on other single-stranded RNA virus also like RSV. So now, after limited clinical studies, we know it is very good on COVID. So now it is approved for human use, and this is one medication that is helping us to mitigate the COVID complications. So if an individual, like an elderly individual or people of high risk, they are slowly moving into the COVID, then this is actually it's being used. It's being available now. Some of the drugs, like say the Regeneron, a company which is actively involved in the development of a couple of drugs, couple of strategies. I won't say drugs, couple of two different strategies. Number one, obviously, being an immunologist, immediately my mind will come, hey, IL-6 is the main culprit. Why not? Because block the IL-6 receptor. Bingo. Yes. If that's what you're thinking, you are a great immunologist, because the minds are thought like that, and that's exactly where the KSCSTE has been developed. It is a monoclonal antibody which actually goes and basically blocks the IL-6 receptor. So obviously, when IL-6 is blocked, then there is no IL-6 driven inflammation. So obviously, you are actually keeping the horse under control. That's great. They also have a second, another product, trying to just immunize and humanize mouse as well as the humans, and try to take that hyperactive or hyperimmune sera, separate antibodies, screen the sera for a highly neutralizing antibodies, and use it as sort of a passive immune therapy. Especially people who are in hospital, then this will be very, very useful, very good, like a passive immune therapy. Vaccines, in the long term, we want to get away from this mask. We want to just reach people, we want to help people, we just want to just be in an environment where we don't have to be basically having social distancing and all those stuff. What is the solution? How we can do that? With a comprehensive vaccine. There are so many different vaccine candidates in different stages of development. Like Moderna has been in a talk for a long time. You know very well, Moderna is basically mRNA based. Take a human mRNA, truncate it, insert the spike protein into the mRNA, and then just package it into a sort of a viral vector, like a vaccine in the individual. mRNA is inserted into the cells. Then mRNA act like a local factory, keep producing more and more of the viral proteins. So the system is constantly primed. Hence, if a real virus is constant, your system is already primed. So it takes care of it. Okay. Still, we are not seeing much data. The company keeps saying it is great, but still there is very little data out in the open domain. So we don't know what exactly the value of it. Sanofi is actually developing a DNA vaccine on a viral backbone, very much similar to what we have discussed just now. But there is the modern technique is a new technique which is not being really just being employed in any existing vaccine. Whereas DNA vaccine, they're all being very well established. And the J&J is actually they are moving forward. They have shown some positive results with the deactivated SARS-CoV-2, which is very similar to what we know about. If you are a vaccinologist, first thing is, okay, this is causing disease, let me take it, kill it or attenuate it and just push it back. So that's the approach, and that seems to be paying way.
Merck has been sort of keeping quiet, keeping silent for a long time, but like in the last 14 days or so, of course, things are going, happening behind the scene. We don't know about anything, but last week we just got a news because I belong to assay development. So if there is going to be a new vaccine molecule, then our group would be reached out to say, like develop like in vitro assays to just like demonstrate or evaluate the vaccine response. So we have been that's the reason why we have been reached out and we have been informed that there is going to be like two different vaccine candidates. I think one basically is I think it's adenovirus based vector based vaccine. So we are just in the process of developing like serological assays, etc. Meanwhile, there is also like one antiviral that's being sort of pursued by Merck, where we are reaching out to a company and buying out a company that is actually having a sort of like an antiviral medication. Obviously, prevention is better than cure. So, best prevention is basically wear a mask, but no need to wear a mask if you are in your own office. So you can basically you can take off the mask and breathe freely. Wash hands. Avoid contact with the people, avoid travel, so stay home, that's very nice. If you are sick, you can stay home. So Google has dedicated 5 rules. Let us just practice these 5 rules so that you can stay safe and others also can stay safe. This is my message. This is my message, like the final slide, is like we are resilient. We have seen so many challenges, natural calamities. We have seen tsunamis. We have seen earthquakes. We have seen endemics, pandemics. We are resilient. We have overcome several challenges. We will overcome this COVID also. But the only thing is, the lesson to learn from this, we need to make sure that we just take efforts to make sure the next pandemic is delayed by several decades. A simple like a lifestyle change. Like, okay, I'm not going to buy any wild animals anymore. I'm not going to just let me just confine my eating habits to sort of sensibly like, okay, I will eat chicken, I will eat mutton, that's fine, but I will just stop with that. I will not try to go for alligator meat. I will not go in for something else like that. So if you are not going to be weird, so you are staying normal, the virus also is going to stay normal, or bacteria also can stay normal. Okay? And now, with the omics explosion, let us try to be proactive. The evolution is going to happen, you cannot stop it. Like a viral virus or bacteria or other parasites, they are going to evolve. But now we have better information about their evolution, the frequency of evolution. So, like young scientists like you who are working on bioinformatics or molecular biology, etc. I think you guys need to come up with modeling, appropriate modeling, a predictive modeling that can basically predict this. If this is what is happening, which could be the next possible threat? Which can be the next possible threat? Can it be a coronavirus? Can it be a virus? Which ones can be the next possible threat? So, what do we do to avoid that threat, or what do we do to just mitigate that threat? So, those I think is possible with modeling. We are doing a reverse modeling. I strongly believe we should also just do a sort of a forecasting modeling so that we can actually be more prepared. More than anything else, I'm telling you, we have a responsibility, especially in the social media. The social media, I'm telling you, it has got both positives and negatives. So much of misinformation is being shared in WhatsApp or Facebook, etc. So much is shared. Please, as a scientist, do a fact check and put an end to it. Or else, people will just go crazy. So, if something comes in your way, like yesterday, I think a few weeks back, I just got a message. One of my colleagues from India saying, hey, looks like a Nobel laureate from Japan said that this virus is constructed in the lab. So, see, look at Nobel laureate himself is saying it. So, it is actually China who has done it. Then basically, when people don't have the patience, okay, I see something, why don't you just Google it? When you just Google it, the same Nobel laureate from Japan, he basically gave a disclaimer that, hey, this is nonsense. I didn't say anything like that. So, but that, it is natural, right? All those negative information gets attraction. Like the vaccine, basically, when you look at the vaccine, the anti-vaccine groups about a possible connection between autism and vaccine. one paper that's came out, which is not before trial, one paper. After that, there are thousands of publications that shows that there is absolutely no connection between the thimerosal, mercury compound, and autism. But still, anti-vaccine groups still persist in their thriving. So it is our responsibility as a scientist and as a part of scientific what is really true and what is not true. Okay, so with that, thank you very much. I really want to thank everybody for this great opportunity. I hope I have given you some information. I know very well I have scratched only the surface. I know that. There are plenty of information. As we talk, I'm pretty sure there are more publications coming out. So I may not have given you a very updated information, but I'm pretty sure I have triggered your interest in COVID so that you will be going back, looking at responsible or like a fact check the social media and actually educate yourself and pass on the information. Thank you very much, and wish you all stay safe.
Thank you so much, sir, for that wonderful lecture. You have covered upon the biology, then the post-immune responses, the updates, all, and we have got a few questions. The participants have posted in the chat box. Shall we look at that, sir? Sir, one Bhanvi Rajam was asking about the vaccine development, sir. So, what about the timeline from the vaccine development to the quick implementation to the public level? That's what she's asking.
I think the way in which companies are actually like for example, I can give you a personal example of Merck. We are basically pretty much like we are just pooling all our resources. What like almost like we're trying to put more like people to work on it. On one hand, FDA is trying to just give fast track the vaccine development. They are not there, so there's no problem in funding. Our hope is at least by mid 2021 we should have a vaccine that can go into the individual. That is very tall call, because usually you know very well how long it takes for the vaccine go through phase 1, establish the safety, then go to phase 2, establish phase 3, establish efficacy in the target groups, then basically maybe phase 3B to see whether it is good in a various geographical region and all. It takes several years. So we are trying to compress and we are hopeful. And it is there are several vaccines which are having a parallel processing. So we hope that at least one vaccine would basically just emerge as a winner within this short period. So I am very hopeful, I'm very positive that we will have a vaccine by the beginning of next year. At least mid next year we will have a vaccine.
Sir, another question is from regarding the herd immunity, sir. What are your thoughts on that, about herd immunity, and will that be effective particularly in the Indian scenario? That's what she's asking.
I think that is what is keeping the infection rate and then fatalities. I wouldn't be surprised that there is homology with several other viruses, and which country among the population, which country has got more exposure to a variety of pathogens. It is only countries like India or African countries. So obviously our immunity, we have an existing immunity for various infections. I am pretty sure that is providing some degree of cross protection. And especially when we have such a condensed population, there is I think one mechanism by which India, I'm pretty sure by the time vaccine is there, I'm pretty sure every Indian would have some degree of antibody level to COVID because of the herd immunity. So I'm pretty sure herd immunity will play a big role in India in protecting the Indian community. Only thing is, let us make sure the high-risk populations are protected. If Islam is protected, we are going to get herd immunity, and that's going to help us.
Yes.
Who knows? Who knows? There's a population already most of them have the herd immunity.
Then what about plasma therapy, sir? What about its efficacy with plasma therapy?
So Then there was communication. Oh, even those who are COVID COVID survivors, they are getting reinfected. So the first the primary infection is not giving enough immunity. Then there were additional research which showed that no, the antibody there is a seroconversion like initially IgM, and there is seroconversion, and those IgGs are quite long-lasting. That's all being demonstrated. Now there is a revived interest in plasma therapy. So plasma therapy certainly has got a value. Certainly has got a value, especially in high-risk patients or hospitalized patients who are really been serious like life support. This will be more like a rescue therapy. Both plasma therapy is more like a rescue therapy, but yes, it has values, and we are getting more and more data on benefit of plasma therapy.
Okay, sir. Then many times the virus will be there in asymptomatic condition also. They'll be the carriers. So one Shabana Mode is asking, what about the destiny of the load in asymptomatic people?
Destiny is your system is taking care of it. Your system is going to knock it off. Hopefully, your system is knocked off. But you are not getting it anymore in the environment. But hey, if I'm asymptomatic, I'm blessed. I'm going to survive.
Yes. Then Dr. Arun is asking, what about what is the calculation for this R0 value, R0 value? Mention, no, sir.
Or not, yeah. That is basically based on like it depends upon how exactly the contagion is actually transmitted, and there is a paper on that. Like in Aruna, if you want, there is a paper on that where it is too much of a. Okay, I'm not a epidemiologist. I can certainly get some information where it can just be like directly into it.
Okay, sir. Then one Soumya is asking, does any droplets are capable of blocking endocytosis can be used for treatment of viral infections?
Why do you want to basically block the endocytosis? What is triggering this? Okay, this is a I don't want to discourage this. So you are in great thought. Ultimately, it is the endocytosis, the process that is actually allowing the virus to just go in. So why not we just block the endocytosis? And that's exactly what we are doing. The vaccines are going to just block that. They are going to block endocytosis by blocking the viral attachment to the by producing neutralizing antibodies. The neutralizing antibodies are going to just block or impede the attachment of virus to the ACE receptors. By blocking the ACE receptor, you are going to block the endocytosis. So you are right. You are pretty much thinking like a vaccinologist. So you can be like a vaccinologist in the future.
A question from Amatira, sir. Now, how can we forecast the upcoming or future pandemic?
That's what I'm telling you. You guys have tools. We have modeling tools. You know exactly what exactly is happening. Basically we know very well every virus or every different virus, we know its replication frequency. We know what is a mutation. We know what is mutation. Like it is a replication error. So it is pretty much like it is calculable. Because basics say like a spontaneous mutation. What is the frequency of spontaneous mutation? Maybe like a one in 10 million or 1 in a million. So we have some definitive number numeric for that. Why not you apply this definitive numeric to a virus? So we know very well, and again we know very well that viruses like during the evolution. Only these viruses have been pre-selected. It's like survival of the fittest. So only those viruses which has got a sort of a constitutive regions, those regions which do not undergo much mutation, and those regions that are hyper mutation spots. So basically, you're not going to just look at the algorithm for the entire genome because virus is not stupid. At least during evolution, what it has happened, it knows that hey, these are I don't want to get mutation in a different place. I'm not stupid. So I want to make sure that the replicase region is actually intact, so the mutation does not happen there. But once a spike protein region, that has got a hypermutation region. So I know very well this is the region. Maybe like for example like a 200 maybe 1,000 base pair or 1,500 base pair. This is the region that's going to undergo mutation. And my question is basically is like, why not we just have an algorithm to say, okay, if it is going to replicate this phase, this is the replication phase. Okay, what is the possibility if I'm running? If I'm running at like a 10 miles per hour, what is the possibility of ice tumbling? If I'm running at 100 miles per hour, what's the possibility of ice tumbling? Obviously, if I'm running at 100 miles per hour, I'm going to stumble faster. If I'm walking, I'm not going to stumble unless I'm really old, I'm really drunk or old man. But if I'm so basically the pace with which you are moving, that is directly it's going to be directly proportional to the possibility of stumbling. Just overlap it with the viral replication. The pace with which it's replicating is going to be directly proportional to the mutation. Then add another factor, the hot spots, the hyperbolic spots where you frequently see the mutation. You have a rich database for the past 10 years, 15 years. Take the database and just make an analysis. It should be possible for us to just come and do some degree of empirical analysis. See, we cannot be taught. So you cannot say, hey, coronavirus, tomorrow you're not going to just infect the you're not going to infect bad anymore. So this tomorrow you are going to generate a new variant that's going to infect, say for example, dolphins. No. But at least we will have some sort of a prediction to say like, hey, there's a possibility that we are going to have maybe a new variant here. And this new variant may happen in spike protein. And we have a three-dimensional docking models now. So if this spike protein is going to change, then where will it dock in the human system? This COVID, even though it is going to be a respiratory virus, the way in which it's docking, it may be possible that it may be a reproductive virus. Because docking seems to be more on the ovary. Oh my gosh, then this fellow can cause a reproductive problem. That sort of a predictions can happen. That's why I feel like science should actually utilize its potential.
And about the virulence, has decreased by time, and also about the possibility of secondary infection. Lakshmi and Ashwati is asking.
Great question. Great question. Secondary infections really that's the one that is really causing a lot of issues. Like when an individual comes down, especially with the respiratory infection, right? When things become bad, even a soft fever can become bad. Like pneumo, almost like every children, not adults, but usually children do carry strep pneumo in the respiratory tract. So if a children gets it, then there's a possibility that like a strep pneumo can actually can overrun the system. On one hand, the immune system, your virus is actually overwhelming your immune system, and at the time when this is happening, it's almost like you are on a steroid. So your immune system is already hijacked by this virus. Hence, you are pretty much naked immunologically, exposing yourself to secondary infection, and yes, they can be a real problem. That's the reason why more there's a strong advocacy of other vaccines to make sure that the individuals are protected from secondary infection, like a pneumonia vaccine, like pneumococcal vaccine. There's a more uptake on that to make sure they are protected in that way.
So also asymptomatic, so still they can be infectious also, right, sir?
Yeah, absolutely. They can keep shedding. They can keep shedding, and if there is going to be any change in their immunological status, they can become symptomatic.
Okay, this question was asked. Yeah, please. So this question was asked by Dr. Shabana, and she's again asking. She's asking this because one of her friend got symptomatic positive and became negative after 3 weeks. But her husband asymptomatically positive, remained positive for last 5 weeks. So
this is a changing scenario where initially the CDC said if you're positive, then even if you're asymptomatic or symptomatic, if you're positive, then you have to just quarantine yourself for 14 days. Now they are advised to quarantine for 28 days.
Yeah.
So
Yes.
it is like it is like what she has seen is real. It's not a diagnostic artifact. Yeah.
Yeah.
So I think again we are learning as the pandemic is progressing. We are learning actually. Initially we thought it would be only like it would be a touch and go. It's the 14 days. Now we realize it is going to be longer. So it is, and again, it may not be that everybody is going to have the 28 days or 25 days. It could be like there are few individuals who may have it. Most may not have, because like any bell-shaped curve, there's always outliers. So there's going to be outliers with no reason. There could be healthy. It could be just a normal individual, but still for some reason they love COVID so much, or COVID seems to love them so much, they just have it for a longer time. They're all good. They're all very good. That's where I just have a research proposal submit to KSCSTE, so they can come in for funding to you.
So Rajam is having a question. He'll ask.
So I don't know whether it is too early to ask this question. So if once the vaccine is out, you told about the scenario, Indian scenario, like we are much more exposed to more kind of viruses, so we have a better immunity. So according to you, do we all need to take the vaccine, like considering all age groups?
I think that is where I'm pretty sure at the time the serological tests will also be very mature. So it'll be, I think it wouldn't hurt if individual just like do a quick serological testing, and if you have got a good antibody titer, then basically like you don't have to bother. But my thing is, given the intensity of this virus, given the severity of this virus, given the nature of this virus being sort of the contagious nature of this virus, if you have have a good herd immunity, it doesn't hurt to just prime it. It doesn't hurt to prime it. So even though you don't really need it, just need it because you have a decent your immunological status is like a decent. Priming with COVID is not going to hurt you. It's only good by improve your immune response. So I would basically say if it is possible, I would certainly would recommend it. And if you're going to be a Merck vaccine, I would strongly recommend.
Because I'm a little skeptical because every year, there's a new disease coming up, we need to take new vaccine every year. So I don't know what all these vaccines are going to do inside my body.
Your system is smart, buddy. Your system is smart. You imagine, like you are, whether you want it or not, we are getting exposed but like what is ubiquitous apart from God? Only microbes are ubiquitous. So we breathe. We are whether you like it or not. You can choose either to just accept a vaccine or not. But as far as the natural infection is concerned, you just cannot escape. There is going to be some of them you are going to just touch and go. Some of them you are not going to just like they're going to be touch and go. And hey, you have like hundreds of millions of T cells, man. Each of them have got a capacity to recognize specific antigen. So your system is smart. Your system is smart. It is not going to produce tons of antibody. It is going to produce an antibody and keep a memory source. That's all. And then what it does, second time when it comes, instead of producing like 1 million antibodies, it is going to produce only 10,000 antibodies, but they are got a very high affinity antibodies. So it goes. If it is going to be a one-time visit by an infective agent, your system may not even want to remember them. It will simply ignore. So our system has got our own fail-safe mechanism. So as far as you are concerned, you don't bother about your immune system. Whatever is, however way you can protect it, you protect it. Whereas how to reset myself, which one to respond, which one to memorize, which one to ignore, our system is fine. So there's going to be, that's the reason why even if there are 20 or 30 different vaccines, we just sort of bombard the kids with so many vaccines, but still, they are immunologically good. They're immunologically strong. So that like, even after all these vaccines, when they get an infection, they just show symptoms and they recover. Most of the time, you don't even need a medication. That is because The system is so smart.
Thanks again, sir, for having accepted our invitation, sir. Thank you so much for having spent so much of time for us. And for the official word of thanks, let me invite Rajam, sir.
Thank you, sir. It was really wonderful, very informative talk on the biology of SARS COVID-19. And on behalf of the council and all the members or the participants, we express our sincere gratitude. And we are so happy to have you on web. And sure, and please do keep in touch, and we will organize further programs. And and on behalf of the KSCSTE, I would like to thank all the participants who have attended the program. And thank you very much. Thanks to our Executive Vice President and Member Secretary for helping us to organize this program. Thank you very much. Thank you.
Thank you, sir.
Pleasure is mine. You all stay safe.
Thank you, sir.
Thank you.